Okay, so I'm going to talk in this video about the lab you guys are going to do this week. So the lab this week is from the Carolina kit. So I hope all of you have purchased this kit because this kit has two experiments. This first experiment we will be doing this week, and second experiment we are going to do around the 7th week. So basically the first week is DNA, RNA and central dogma. So in this lab basically you have to make some models, and you have to basically there are 5 activities. The first activity is you have to make the structure of DNA. Then you have to see how does DNA replicates, and then you will be doing an activity on transcription, translation, and last thing is about the mutations. So in this video I'm going to give you some instructions and also going to give you some background. So let's have the background first. So DNA, I think I have talked in detail in my lectures about the structure of DNA. You know what DNA is, how it is made. It's a polymer, it's made of nucleotides. You know the structure of nucleotide. So you know the structure of DNA. So second thing which we need to know is the DNA replication. DNA replication also we have talked, but I would like to tell you one more thing in this. So basically if you have already seen my second lecture, I have told you a little bit in more detail about the how does DNA replicates. So basically by now we know DNA is double stranded. For the DNA to make its copy, the two strands of DNA they have to separate, and each strand is going to act as a parent strand, which means whatever is the sequence on that DNA strand is going to be used for making a complementary daughter strand. So that complementary daughter strand is going to be formed with the help of the enzyme DNA polymerase. And also we discussed in lecture number 2 that DNA polymerase, it adds nucleotides in 5' to 3' direction. So basically DNA polymerase, remember that it's very important, DNA polymerase is going to add nucleotides in 5' to 3' direction, which means that it is going to add a nucleotide at the free 3' end. That in the deoxynucleotides, the functional group on 3' is hydroxyl group, OH group. So there is a limitation of this DNA polymerase that it cannot initiate the process, it can elongate. So DNA polymerase cannot initiate on its own, so it needs a primer. So in the sequencing method we saw the primer was DNA, but at that time I told you in our body, in our cells, the primer is not made of DNA, the primer is made of RNA. So it's a RNA primer which is used in the process of replication, and the free 3' end of the last ribonucleotide will be used by DNA polymerase to add the nucleotides, and those nucleotides will be added on the basis of complementary base pairing rule. So whatever nucleotide is on DNA strand, suppose it's A, T will be added. If it is G, C will be added. And also another important thing I would like to add at this point is, as you know, DNA is made of 2 strands, and those 2 strands are antiparallel in nature. Okay, the 2 strands are antiparallel in nature. One strand is from 5' to 3'. Let me see if we can have a picture here. Okay, just this is a figure for transcription. This is a figure for transcription, but I'm going to use this to explain you the replication. So you can see here, these are the 2 strands of DNA. The 2 strands of DNA, they are antiparallel in nature. If this is the 3' and the other one will be 5'. On the other strand, if this is 5', this is 3'. If this is 3', it's going to be 5'. I just said DNA polymerase can add nucleotides only in 5' to 3' direction. Okay, it can add nucleotides only to 5' to 3' direction. So if you see this strand, this is 3', this is 5 prime. If the primer is attached on this side, okay, primer is a RNA in the cells, but in the labs when we do these reactions, we use our DNA as a primer. So if this is 3 prime, this is 5 prime. If the primer's direction is from here to here, which means it will be 5 prime on this side, 3 prime on this side. So if this is 5 prime, this is 3 prime, DNA polymerase can go and keep on adding nucleotides, and this strand will be made. This strand is called a leading strand because this strand can be made continuously. But if you see the opposite strand, this strand, okay, this is the 5 prime. If you have a primer here, that primer's orientation will be, here will be 3 prime and this side will be the 5 prime. Now DNA polymerase cannot add nucleotides at the 5 prime end. So how this strand is going to be made? This strand is going to be made in small fragments, okay. So it will start, maybe it can be a small primer here which is in 5 prime to 3 prime direction. So it will be made in this direction, okay, and the opposite strand is made in fragments, and that strand is called a lagging strand. So in this strand there will be a small primer here which will be 5 prime to 3 prime. So this strand will be having multiple primers. Suppose one primer is here, second primer is here, third primer is here. So so that primer will be 5 prime to 3 prime. So the DNA polymerase can add the DNA here. Then again this is 5 prime to 3 prime, it will add here. So the key thing is DNA polymerase needs a 3 prime end. So the important point to remember here is, in the DNA replication, one strand is made continuously. That strand is called the leading strand, and the opposite strand is made discontinuously. It is made in the fragments. That strand is called the lagging strand, and this is all because of the antiparallel nature of the two strands of DNA. Another important thing I would like to tell you guys here is how does DNA replicates itself. Mode of replication is called semi-conservative mode of replication. It's called semi-conservative mode of replication because from one molecule of DNA, when two molecules of DNA are going to be formed, in both the molecules of DNA, both daughter molecules, one strand is going to be the old strand and one strand is going to be the new strand. That is why it's called a semi-conservative mode of replication. Okay, so this was replication. In the transcription, basically I have already explained about transcription in lecture number 1. If you want, you can go back to those notes also. So basically in transcription, from the sequence, from the knowledge, whatever is the information in DNA, RNA is made and it is called mRNA, and this is made with the help of another enzyme called RNA polymerase. The enzyme involved in DNA replication is DNA polymerase. The enzyme involved in transcription is RNA polymerase. So these are the two strands of DNA. The two strands will separate, okay, and one of the strand, out of the two strands in transcription, only one strand is going to be read. So this is the 3 prime, this is the 5 prime. So this will be the 5 prime side and this will be the 3 prime. So another thing again very important is this RNA polymerase. So right now I'm talking about transcription. RNA polymerase also adds ribonucleotides, okay. So the function of RNA polymerase is to make a RNA, okay. So it is going to add the ribonucleotides, and it is also going to add the ribonucleotides on the basis of complementary base pairing rules, and it is also going to add the nucleotides from 5 prime to 3 prime direction, okay. Again, so replication, transcription and even the translation, they all take place in 5 prime to 3 prime direction. So there are two strands of DNA, only one strand is going to be read. See, this is the strand which is 3 prime to 5 prime. So the ribonucleotide will be made, this RNA transcript will be made from 5' to 3' direction. So this RNA polymerase is going to go and read the DNA strand and make a RNA mRNA on the basis of the sequence on the DNA. If it is A in the DNA, it is going to add U because there is no T in RNA. If it is T here, it is going to add A because A is there. If it is T, it's going to add A again. So if it is A, it's going to add U. If it is T, it is going to add A. If it is G, it's going to add C. If it is A, it's going to add U. So it is again the same complementary base pairing rules. The only difference is instead of T in RNA, there is no T, there is uracil. And this is the way your mRNA is going to be made. And again remember, RNA polymerase add ribonucleotides in 5' to 3' direction. So after the transcription, so when the mRNA is formed, this mRNA goes out of the nucleus and it reaches the cytoplasm, where on the ribosomes, the protein factories, the polypeptide chains, they're going to be made, and they are made with the help of this another kind of RNA which is called tRNA. tRNA is transfer RNA. Now what is the structure of transfer RNA? Remember that it is again single stranded, but there can be the formation of some loops. Now the structure of tRNA is such that there are two ends. On one end, an amino acid is attached. On the other end is an anticodon. If you remember from lecture number 1, what is a codon? Remember that codon is present on mRNA. mRNA which is formed by the process of transcription from DNA, that is mRNA. And the 3 ribonucleotides of the mRNA forms one codon, and every codon codes for one amino acid. And how does it codes? It codes through the help of tRNA, because this is the structure of tRNA. On one end it has a anticodon, other end it has a amino acid binding site. So whatever is the codon here, suppose like GGC, it codes for amino acid serine. Okay, so which means that amino acid will be brought by this tRNA. So it has to has a specific anticodon. And what is the anticodon? It is complementary to the codon. If suppose it is GGC, which means the anticodon on tRNA will be CGG, and then means it will be having here GCC, and if the codon on mRNA is GGC, the anticodon will be CCG, and on this side it will be having a serine. So that's the way it is. So this tRNA has an anticodon, and this anticodon recognizes a codon. Codon is on mRNA, anticodon is on tRNA, and on the basis of the codon, there is a in this exercise only, there is one genetic code table given in which you can see the various codons which code for which amino acid. So here is the genetic code table. Here basically you can see here the all information is given. It's even in the lecture which codon codes for which amino acid. But how does it actually codes? It codes through this tRNA. So you can see here these are the various codons, like AUG, which is always a start codon. It also codes for amino acid methionine. AUG is your codon number 1. Then ACG is your codon number 2. It codes for threonine. Then we have GAU code is codon number 3. It codes for this aspartic acid. So you can see here these codons, they code for amino acids, but how these amino acids are recruited? They are recruited through tRNA, and tRNA recognizes the codon through its anticodon. So this is all basically about the translation you need to know. The last activity is on mutation. Now, what is a mutation? Mutation is a sudden change in the sequence of DNA, which means either some nitrogenous base can be deleted, some can be inserted, or some can be substituted. So if there is a mutation, which is a change in the sequence of DNA, it is going to, so any change in DNA is going to affect mRNA, and any change in mRNA is going to affect the protein. Any change in mRNA is going to affect the amino acid, and the sequence of amino acids makes a polypeptide chain, and polypeptide chains together form the protein. So mutations, basically mutation, a change in DNA is going to affect the protein. So there will be there are actually 5 activities. Let's go over. So this was all background information I wanted to give you about all these activities you're going to do. So basically this is a kit in which you have been given various beads. So the white beads, they represent deoxyribose, which means this is the pentose sugar which is present in DNA. Then red beads, they represent phosphate group. Then there are orange beads which are adenine, green beads guanine, blue ones cytosine, yellow ones thymine, purple ones are uracil, pink beads are your ribose sugar which is present in RNA, and these clear connectors are your hydrogen bonds. They will help in connecting your strands of DNA. So what you have to do in these activities, in the activity number 1, you have to make a model of DNA. So you have to basically making the help of making use of these beads, you have to you will be making the model of DNA. You are going to connect all these beads, the red ones, the white ones, and whatever the nucleotide sequence is given to you. On the basis of the activity, you are going to make the model of your DNA, and remember that the model you have to make 2 strands of DNA. And after that, once your model is made, click the picture. So I have provided the 5 questions, and this assignment is worth 10 points. Every question is worth 2 points. Once your model is made, just click a picture, and then in the Word document just insert that picture so that I can see you actually did it, and I would also like to see if your model is correct or not. The second model is basically the DNA structure you have made. It has 2 strands. You are going to do with the same DNA molecule, you can do the replication. So you're going to separate those 2 strands, and then you're going to make the daughter strands. So the key thing will be you have to make the RNA primers. For the leading strand there will be only 1 primer, and for the lagging strand there can be few primers. You can go over the instructions and do the way you have to do. It's very straightforward, very straightforward. And you need to know that the mode of DNA replication is semi-conservative. I think there is a question in worksheet where you have to comment on the semi-conservative mode of replication. So you will be actually, when you will be doing with your own hands, you will be able to understand actually how DNA replicates. You will be able to comment on that question. And third is about the transcription. Here I think a sequence for your activity number 3, there has been given, this is the DNA sequence which has been given to you. You will be making this DNA sequence, you will be making with these beads, and then you have to construct a mRNA. So because if you know the DNA sequence, you have to make a RNA sequence. And remember that when you are making a RNA, you have to use the sugar ribose, the pink ones, and you have to use instead of thymine, you have to use uracil. And also once you're doing that, there's a table here. You can fill like for activity number 3, 4 and 5, they have provided your table. You can write down your sequences here. This you don't need to send it to me because this is not going to be graded. The grading will be done on that worksheet. So you can, when you are doing this, is for your convenience, that you can write down the things, and then on the basis of this table you have to answer the questions in the worksheet. So for activity number 3, you have to tell the direction. How does the RNA polymerase it adds the ribonucleotides? You have to comment on that. And activity number 4 is on the basis of the mRNA you have synthesized, you have made in activity number 3, you have to make the polypeptide. So for that you have to see your codons. First of all, you have to recognize your codons, and then for those codons you have to go in this table and you have to find which codon codes for which amino acid, and on the basis of that you can add your amino acids and make a polypeptide chain. And again in the table you can write down your data. This is the amino acid sequence, you can write down here. And I think the question number 4 in your worksheet is asking you about the amino acid sequence. You have to write down the amino acid sequence in question number 4. And your activity number 5 is about mutations. You have to make some changes in your DNA, and you have to first of all make your DNA strand. I think this is the DNA strand, and then you have to make some point mutations in your DNA, and then see when your DNA is changed, how does it affect mRNA, and when your mRNA is affected, how does it affect your protein. And again you can write down here your mutated DNA sequence, your RNA complement, and your amino acid sequence. This is very straightforward, it's very self-explanatory, the information is there in this kit. But still, if you have any question, you are very welcome to talk to me. You can do this activity. This activity will actually make you very clear about how these processes takes place, and you will be able to get a good hands-on experience. And I hope you will understand very nicely all these mechanisms. You will get a hold on all these mechanisms: transcription, translation, and your if there is any kind of a mutation, what happens.